Right, today we'll look at microbial metabolism. So, what exactly is metabolism? It's a umbrella term that kind of describes all the different chemical reactions that take place within an organism, usually in reference to either breaking things down or building things up within the organism. And colloquially, so just as a general term, kind of casually, we use it to refer to how organisms break down the nutrients that they either ingest or create themselves in order to obtain energy. So we can classify the different types of metabolism based on first we'll look at the inputs and later we'll look at the outputs. So here we're looking at the first energy source, electron source, and then third carbon source. These are the things that are going to go into the organism, and the organism will use this to create the energy, right? And this electron source, I'll describe exactly why this is necessary in the coming few slides. And this carbon source is the same thing as think of like sugars or like glucose, for example, right? Glucose is the main thing that our bodies need. So, yeah. Oh, I guess I already highlighted this. But yeah, so for if we look at energy source, we can classify organisms into either phototrophs or chemotrophs. So a phototroph will obtain energy from light, whereas chemotrophs, they obtain energy from chemical compounds that usually they can either acquire through, like getting them from the environment, or they could consume an organism and take those chemical compounds, etc. And then for the electron source, either you get them from organic compounds, which contain carbon and hydrogen bonds like this, whereas lithotrophs, they will obtain electrons from inorganic compounds, such as metals or different metal ions. And then for looking at the carbon source, finally, carbon/sugar source, either these are probably the ones you've heard of the most. Either you're an autotroph, where you can make your own glucose/carbon, often through photosynthesis, such as the way that plants acquire their glucose, or you can consume other organisms for that glucose. So you have these three different categories. Basically, you choose one from each quote unquote bucket here, and then you can describe how the metabolism of a certain organism works. So you can see energy, electron, carbon source. You combine the three, you make this long, really long prefix here. So photo, organo, heterotroph, that would mean you get energy from light, you get electrons from organic compounds, and you also consume other organisms. So you can see all the different categorizations here, and you can see that some of them don't have any organisms here. But just good to be familiar with all that nomenclature. So now let's look at like energy and carbon sources, that kind of makes sense. But why do we need an electron source to be able to perform metabolism? And what does that really mean? So most of the reactions, right? Like as you can see here, this is like a metabolic pathway, right? So you can see how there's a very complex system here, made of various enzymes, and then even like tens to hundreds of different chemical reactions that take place just to break down, for example, one glucose molecule. So, and it turns out that a lot of the reactions in this pathway are what are called redox reactions. And redox reactions, they stand for reduction oxidation reactions, and they involve some sort of transfer of electrons. And I mean, you might think, right, like all chemical reactions involve transfer of electrons, but this is slightly different because it involves a change in what's called the oxidation number of the two things that are reacting together. So we're not going to get into the chemistry here, but just know that these redox reactions are a very particular type of reaction that involve electron transfer. And since we need to transfer these electrons, you can kind of think of it as we need an electron source to kind of push or drive those reactions forward. So we need some kind of source to acquire these electrons. And briefly looking at redox reactions, essentially there's one, so species just means one of the reacting components. So one species is going to be reduced while the other is oxidized here. So reduction essentially means that or you can look at this, what's this called? Mnemonic, right? Yeah. So oil rig. So reduction is gain, and then oxidation is loss. And this is referring to electrons. So if you're reduced, you're going to take in electrons or get them from somewhere. Whereas if you're oxidized, then you're going to give away your electrons. This will come in handy later when we talk about some basic oxidation reduction stuff. So, for example, if you look at the, this is not a reaction that would take place in the body, right? But this is something that happens in an electrode or a battery. So you have zinc, you go from zinc neutral to zinc 2 plus, which means that you lost electrons, right? Because you're becoming more positive. Whereas the Cu, the copper, goes from 2 plus to neutral, which means that it had to gain electrons. So it gained negative to kind of neutralize this positive and make it neutral again. So, yeah. And then I would recommend before, so I have like I briefly cover like, you know, respiration, fermentation, photosynthesis, etc. And I put these links to review videos. I already posted this on the Google Classroom, but in case you didn't get a chance to watch, I would recommend watching this entire video before moving on to the next component here. But I mean, we're not going to do that now. So after you watch that video, I'm going to assume you just watched the video. So just to recap, right, this is the overall equation for cellular respiration, and this is the same between prokaryotes and eukaryotes. So we have glucose and 6 oxygen molecules, and this is what's called an oxidation event, and then that causes, or that I guess creates as a byproduct 6 CO2 and 6 water here. Yeah, so in eukaryotes, right, we talked about how, or I mean, I believe it was discussed in the video, but glycolysis, right, the first step where we're breaking down the glucose into pyruvate, right, that takes place within the cytoplasm whenever that glucose is transported into the cell, whereas the next two components of respiration, the Krebs cycle and the electron transport chain, these are actually localized within the mitochondria. And specifically, right, the electron transport chain, it's embedded within the inner membrane of the mitochondria. As you can see, we zoomed in on this component here. And we can see this inner membrane contains the ETC, and the H plus, right, the protons are going to be pumped from the matrix, well, not necessarily from the matrix, but, you know, out of the matrix towards the intermembrane space between the two membranes, and they're going to go back through ATP synthase, which is kind of like a water wheel. This water wheel is turning and generating ATP from here, and it goes back into that mitochondrial matrix. But, you know, the thing is, obviously, prokaryotes don't have mitochondria. In fact, they are where mitochondria actually came from. So that means that, right, then where are the Krebs cycle and the electron transport chain going to occur? So glycolysis, the same. It occurs as the glucose is imported into the cell in the cytoplasm. And actually I'll write this down. I think I forgot to write it here. Yes. And this includes Krebs. So the Krebs cycle will also take place within the cell, just the cytoplasm here. And now the electron transport chain is actually going to be localized on the membrane itself of the bacterium, which makes sense because, of course, the mitochondria was a bacterium and it has the ETC on its membrane. So the bacteria should have the ETC on its actual membrane as well. And here, it's kind of the same logic as here, right? The H plus is going to get pushed outside of the cell, and then it can enter back in through this ATP synthase complex and then generate ATP. And one thing that's different here is that this is called the proton motive force, this forced flow of protons due to an electrochemical gradient, right? And this is going to also be used to drive other processes such as flagellum rotation. So, right, I run over this a little bit in my bacterial structure lecture, right? But some of these H pluses will actually go back in and pass through the motor complex of a flagellum and drive that rotation so that the flagellum can allow the bacterium to swim as well. Okay, and again, fermentation review video, go ahead and watch this before looking at the next few slides. So yeah, fermentation is, going over the key points, fermentation is anaerobic, so it is, I guess, happens preferentially in the presence of I said that very weird, but with no oxygen present. So we'll talk about this concept of the terminal electron acceptor. So, right? In the ETC, you had that chain of proteins. We'll say this is the chain of proteins. The electrons are passed. And then remember, we needed O2 at the end to kind of carry away all these extra electrons that came from the electron transport chain. So oxygen is what's called the terminal electron acceptor. So it's accepting all these electrons. And it's carrying them away because otherwise, right, if we get a bunch of electrons just kind of clogging up the system here, then the system won't be able to function as normal. So when oxygen is depleted, that's exactly what happens. So we don't have something to take away the electrons. This whole system gets backed up, right? And then we'll see what happens there. So since the electrons can't be carried away by the oxygen, right? And I believe this should be in the video, but yeah, so the electrons are instead, they're kind of shoved back onto the pyruvates that were created from the glycolysis event. And then that can either convert them to ethanol and CO2 or to lactic acid. So we have two types of fermentation here. Either in the absence of oxygen, right, it's either going to produce, the organism will either produce ethanol or produce lactic acid. And this should also be mentioned in the video, but fermentation, right, it produces 2 ATP approximately, whereas normal respiration with oxygen produces 30 to 38. So you can see it's much less efficient, and it also produces these kind of side products that aren't always ideal. And then the sugar sources, right? We've gone over, right, in different in the what was it called? The bacterial culture lecture, I believe, where we said that, you know, different bacteria are able to ferment different sugar sources. So those include glucose, lactose, sucrose, etc. So as an overall view, fermentation starts with some sugar source and a lack of oxygen, and it then results in either the production of ethanol, which is just alcohol plus carbon dioxide, or lactic acid, as well as minimal ATP for the organism. And we'll see that these processes can be harnessed for food production and other applications. Oh, yes, and this is also good. So fermentation occurs exclusively in the cytoplasm because remember, right, if we're looking at an eukaryotic organism here, then the ETC and the Krebs cycle are both occurring in the mitochondria. So both of those processes are backed up, and we can only do glycolysis here and then kind of shove these electrons back onto the pyruvates, right? So fermentation is only occurring within the cytoplasm in eukaryotes. And then for prokaryotes, everything's occurring in the cytoplasm anyway, so it doesn't really matter. Okay, and then let's look at this process called anaerobic respiration. So this is a different thing from fermentation. Because fermentation, we're not actually doing the full respiration, right? We're kind of stuck at the first part of respiration, and we're kind of just like cycling back through it. But here, some microbes, right, they're completely anaerobic. They're obligate anaerobes, which means that they live, like they will die if there's oxygen present. So they have to live in places with zero oxygen. And of course, you wouldn't expect them to only do fermentation for their entire life, right? Because the ATP yield is so low, as we saw here. We only get 2 versus 30 to 38. So how do they get their energy? So instead of using oxygen as the terminal electron acceptor, they do the whole cellular respiration thing, except they just use something else to take away the electrons at the end of the process. And this is what's called anaerobic respiration, again, emphasizing that it's not including fermentation here. And we'll get into what exactly these terminal electron acceptors can be. So remember at the very start, we talked about electron sources and the different ways, either like, chemotroph or lithotroph, right? Let me go back here. Or sorry, organotroph or lithotroph, right? Those you can either get your electrons from organic compounds or inorganic compounds. And so there we talked about the electron sources, and now we just talked about the different terminal electron acceptors. So if we really, really simplify down respiration, we can view it as it's starting with an electron source, passing those electrons through various redox reactions, and finally those electrons arriving at an electron acceptor that takes away those electrons, and then ATP will be created in the process. So very simplified view of respiration. But this is useful because that means we can now categorize organisms into something, an X oxidizer and a Y reducer. So what does this mean? So this electron source is the X here. I'm just using variables as stand-ins, and this acceptor is the Y. So remember, when something's oxidized, it's going to lose electrons, right? So our electron source is getting oxidized because it's giving away these electrons to use for all the reactions within the process here. Whereas this reduction, right, reduction equals gain of electrons, this electron acceptor is gaining electrons that have passed through the process and taking them away. So this source is oxidized and the acceptor is reduced. So we can say, for example, we can say an organism is a sulfur compound oxidizer, which means that it uses sulfur compounds it extracts electrons from sulfur compounds and uses them for respiration. You can also say it's a iron 2 plus oxidizer, which means that it uses iron 2 plus for electrons. Then we also have various examples of alternate electron acceptors other than oxygen. So if we say something is this is sulfate, right? So sulfate reducer, it means sulfate is the terminal electron acceptor. Whereas if something is a ferric ion or Fe3 plus reducer, that means it uses this guy to carry away the electrons at the end of the respiration process. And a good thing to note is that oxygen is the best electron acceptor, which means that it makes the process most efficient. So using oxygen produces the most ATP, but since these other alternate microbes have grown for like millions of years in conditions without oxygen, right, they've evolved slightly less efficient systems that still get the job done here. And we'll discuss, right, there's this thing called Winogradsky columns. It's where you can grow a ton of different types of microbes that use all of these different types of metabolism styles in one kind of container. And we'll discuss that more as we get on to the state national topics. And then, okay, now let's move on to photosynthesis. We have, again, I have a review video to watch before looking at the rest of these slides. So hopefully you watch that and you get that photosynthesis is long and complex. So I'll just go over the main points here. So one of the most important things is the overall reaction for photosynthesis, which involves 6 CO2, 6 water, and also we also need, right, sunlight. Let me try if I can draw this. We need sunlight kind of to activate this process with energy, and we get glucose plus oxygen. And we also technically we need electrons, right? Because the sunlight is actually exciting the electrons and causing them to move down the first electron transport chain at photosystem 2. Anyways. Okay, so there's two types of photosynthesis. The first is oxygenic photosynthesis, which refers to the photosynthesis that we're all kind of familiar with, where, right, we have the process called photolysis, where water is split apart to provide both electrons and protons for the electron transport chain. And as a result of that, we get oxygen production as well. And this is what most plants, algae, cyanobacteria, etc., do to create glucose. It's the thing that we've all learned about in biology class here. Then we also have anoxygenic photosynthesis, which, as you can imagine, is an alternate form that doesn't release O2. So now, instead of splitting water into electrons, protons, and oxygen, now these specific types of microbes use different compounds. So for example, there's this class of bacteria called green sulfur bacteria. Instead of water, they use hydrogen sulfide. You can see it's pretty similar. Instead of H2O, we have H2S, right? So so they use H2S, they split this, they get electrons and protons, and they get, of course, S, this elemental sulfur released as a byproduct. And this is also involved in various nutrient cycling. So when sulfur is moving through the environment in various different forms and cycling around, it's also involved in that. So. Okay, nitrogen fixation. So this one will be a new one. So nitrogen fixation, also called diazotrophy. This is a process by which certain microbes can convert diatomic nitrogen in the air, right, N2 floating around in the air, and our atmosphere is actually around 70% nitrogen, and they can convert them into ammonia, or NH3. And why this is important is because there's something called the nitrogen cycle, where nitrogen is basically cycled through the environment in various different life forms, in different chemical compounds that contain nitrogen. So you can see, for example, nitrogen fixation is particularly important here, within the roots of different plants. But you can also see that there's various other compounds like ammonium, nitrites, nitrates, and then all this other stuff that kind of cycles the nitrogen through the soil and into different plants, back to the atmosphere, etc. So, and the organisms that can perform this conversion of N2 to NH3 are called diazotrophs. This process, we're not going to get into all the molecular mechanisms of this process as we did with like respiration or photosynthesis in those videos, in the review videos. But essentially this process, it's pretty energy intensive. So around 12 ATP, just because this nitrogen, right, the N2 actually has a triple bond between it, which I couldn't really draw on the slides here, but yeah, there's 3 bonds between the nitrogen. Which is kind of atypical because, like, for example, like hydrogen, right, H2 has 1 bond, and then O2 has like 2 bonds, but N2 has 3 bonds between it. So it's a very difficult bond to break, which means it requires a lot of energy, and it does that by using the nitrogenase enzyme to catalyze the process along with a lot of ATP here. Yeah, so the reason why this is important is because, right, N2 floating around in the air is not able to be captured by most plants. But the plants can absorb ammonia that is present within the soil. And remember, the bacteria are converting the nitrogen from the air into NH3 in the soil. So these bacteria are basically doing the job for them and giving making the food available to the plants, essentially. So you can imagine there's some sort of symbiotic relationship that's occurring between these bacteria that can do nitrogen fixation and plants that can utilize ammonia for their cellular processes. And the most notable example of this is between legume plants. These include beans, chickpeas, peanuts, essentially anything that kind of looks like a bean plant. And also this specific group of bacteria, which are known as rhizobia. And these are they kind of live on the roots of these plants, and they're also diazotrophic, as we've been discussing, so they can do nitrogen fixation. So these rhizobia, they are attracted to certain compounds that are produced by the roots of these legume plants, and then they secrete what are called nod factors. And these are just proteins that kind of induce the formation of what are called nodules on the plant roots. So whenever the plant roots come in contact with nod factors, they form these kind of extensions or kind of swelling up of the roots here, and then those will come to house the new rhizobia bacteria that have, I guess, came in and quote unquote infected the plant. So the rhizobia, like we said, will convert the diatomic nitrogen into ammonia to quote-unquote feed the plant, while the plant can also provide energy and protection for the bacteria that are within the nodule. So this is a mutualistic relationship where both of the parties benefit. And we'll get into more types of these symbiotic relationships in a future lecture about microbial ecology.